So what do we want to do today? We want to make sure that by the end of the lesson, you're able to understand the evidence that led to the law of independent assortment. Can you remember that? What independent assortment was? Well, hopefully we'll recap it. And to be able to predict the genotypes from the phenotypic ratios of the offspring. So you will be working backwards because, of course, it would be too easy to just work forwards. Okay? You might get questions that ask you to work forwards, but mostly you're going to be asked to be looking for specific ratios that make you work backwards. So if we just go through some of the words that we've used so far, we've used monohybrid inheritance, and that's when you've got one gene on one pair of chromosomes. It might have 2 alleles, it might have 3 alleles or whatever, 3 types of alleles, but you've just got one gene on one pair of alleles. And that's what we've always considered, isn't it? When we've talked about brown eyes, blue eyes, whatever, we've never known anything different. What we've been talking about is monohybrid inheritance. Dihybrid is when you've got 2 genes on 2 different pairs of chromosomes. Now that is different.
Same genes?
No, so you might have brown eyes and blue eyes, which is one gene with 2 alleles on one pair of chromosomes, and then on another pair of chromosomes you might have curly hair and straight hair. Okay, so you've got 2 sets of chromosomes. And the reason why I mention this one, autosomal inheritance, which we come to later, is because that's when we have 2 pairs of genes on one pair of chromosomes. So let me explain further. Okay, this is an example here of what we know and love. That's what we've been doing since GCSE, right? You've got one pair of chromosomes and there is your gene. This is what we're looking at today, which is dihybrid inheritance. You've got 2 pairs of chromosomes. I can tell that's 2 pairs of chromosomes because that's a homologous pair and that's a homologous pair because they're about the same size. So this is dihybrid inheritance. I've got a gene there on one pair and I've got a gene there on another pair. Could be eye colour, could be curly hair, straight hair. On this one, which is autosomal linkage, which comes on in a couple of lessons, and I think the reason why it comes on in a couple of lessons is because if you did it straight after dihybrid inheritance, it can get a bit confusing. So after this lesson, we move on to codominance and a few other bits and bobs, and we come back to that, and that is slightly different again. So this is what we're focusing on today. And the whole point of it is, because again with AQA they want to know about evidence, is how coming up with this 9:3 to 3:1 ratio led to the evidence, led to the idea and provided the evidence behind the idea of independent assortment. Just to remind us, the law of independent assortment means that if these were making gametes, you've got your 2 chromosomes, and of course we know that in a gamete you would have half the number of chromosomes and you would have 1 of each homologous pair. And so what we're saying is that this one can go here and it could join with this one. Okay, I'll change colour just because we can. So let's go to this one. This one could join with this one. Change colour again. This one could join with this one. And this one could join with this one. So they can combine in lots of different ways. Doesn't mean that this one is going to combine with this one and this one's going to combine with this one. It might do, but it might not. It's the independent law of assortment. Let's consider then something really boring like pea plants, because they never give you anything particularly interesting. They're never going to give you anything that is going to tickle your fancy, as we would say. So if we've got pea plants, let's imagine that we've got green and yellow types of plant. So we're going to let the big G equal green, the small g, obviously our recessive, equal yellow. And we're going to let R equal round, which is dominant, to the recessive, which is small r, which is wrinkled. Okay, and we're going to get a pure breed times a pure breed. Okay, so if we have a little thing, the phenotype is going to be green and round, and I'm just telling you that the genotype would be big G, big R, big R. It could actually have been big G, big R, little r, couldn't it? Well, I'm telling you that. And then equally, we've got yellow and wrinkled, and that would have to be the genotype. So we're now looking at what gametes, if we cross those plants, what gametes we could produce. So this one, the only gamete it can produce would be a G and an R, because it doesn't matter, like with the previous diagram, how they combine, that's all that can arise from that. And to show that it's a gamete, we're going to show it that it's a discrete unit, and we're going to put a circle around it. Okay? And then this one, all that this one can form is a little g and a little r. Agreed? Now when I put those in a Punnett diagram, I don't need to go to any great lengths, because of course it's a very small diagram, because there's my gamete there, there's my gamete there, and so the only offspring are going to have the same phenotype and the same genotype. It's going to be heterozygous, it's going to be green, and it's going to be round, okay? So that is my F1 generation. Agreed? Okay, so let's have a look now, when we cross the F1 generation, what happens next? I'll come over here actually. So what we're looking at now is we've got our F1 generation, and we know that all the seeds in our F1 generation have that genotype. Okay, so what gametes can they make? Well, this G can combine with this R, so we can have GR. This G can combine with this R, so we can have that. This G can combine with this, and this G can combine with this. Agreed? And if we're crossing that with more of the same, because it's the F1 generation with the F1 generation, I'm going to have exactly the same down the other side. Yeah? So I'm going to have GR, 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 GR. Okay, and now I'm going to work out what would be the genotypes of the F2 offspring, because if you remember, this is already the F1. So I'm going to have G. Okay, so now let's just consider what different phenotypes I've got, what is physically expressed. So the possible combinations I can have are green and round, I can have green and wrinkled, I can have yellow and round, and I can have yellow and wrinkled. And I think you know where we are going with this, okay? So let's have a little look. This one would be green and round, okay? Green and round, green and round, green and round, green and round, green and wrinkled, green and round, green and wrinkled, green and round, green and round, yellow and round, yellow and round, green and round, green and wrinkled, yellow and round. So now what we've got is a 9:3:3:1 ratio. and that is indicative of the F2 generation. When you've had a pure breed and a pure breed, you've crossed them, you've got your F1 generation. You take your F1 generation, which is heterozygous, for both alleles, for both genes, and what you're then going to get is this. So where's that going to go in an AQA question? Okay, in an AQA question, they're going to give you a question, and it will be something like, someone grew some seeds, and they found that 279 of them were green and round seeds. They found that 96 of them were green and wrinkled, and they found that 101 of them were yellow and round, and they found that 32 of them were yellow and wrinkled. And they could easily ask you, what they're expecting you to do is click on that here we've roughly got a 9 to 3 to 3 to 1 ratio. So from that, whatever the example is, you know that you've got a heterozygous times a heterozygous. And the generation before that was homozygous versus the homozygous. And that is your dihybrid cross. And that, and the experiments that Mendel did on his pea plants, clearly a guy with a lot of time on his hands, spent a lot of time on his own in that greenhouse, he came up with this rule that it's independent assortment, that any one of the chromosomes of one pair can combine with any one of the chromosomes of another pair. Now to us, that's dead simple, because we're like, yeah, someone else has worked it out. It's always really easy when someone else has worked it out. And not only that, we're just looking at 2 pairs of chromosomes. Well, obviously, if you imagine the number of varieties that can take place, with the many chromosomes and the many genes on those chromosomes, it is really quite incredible that he worked it out. So we will give him a little bit of credit for it, and that's why it's gone down, not just in the history books, but in our science books too. So that is dihybrid inheritance, a step on from monohybrid and a little bit further towards autosomal linkage, which we're not talking about today.